Hi everyone, my name is Hailey Crusenberry and thank you so much for your consideration for this scholarship. Today I'm here to tell you a little bit about our National Science Foundation undergraduate research. In our lab, we do directed evolution of a cytochrome P450 enzyme for the synthesis of marine natural products. Now, as scientists, we all know the general definition of an enzyme. It lowers the activation energy needed for a reaction to take place, typically making it occur at a faster rate or more efficiently. In our case, our enzyme is our BMP7 cytochrome P450. Now we know once we express this, we can put it into a reaction with the protein produced. So we have our substrates, which are brominated in this case, and we know that these attach to the heme binding site at the center of that enzyme with the iron core. And hopefully this gives us our desired product, which is hinokial. Hinokial is an anti-inflammatory and anti-cancer product that normally through chemical synthesis means is very difficult to form due to the covalent bond highlighted in red. So we're trying to make our lives a little bit easier. Now this summer we decided to do a carbon monoxide binding assay to make sure our research was accurate. We can tell two things. One, that our heme is active due to the peak at 450 nanometers, versus if it was inactive, it would be at 420 nanometers. And we can, too, tell that our substrate is binding accurately because of the slight but visual horizontal shift in our graph. Now, when this heme is active at that binding site, we know this catalytic regeneration cycle takes place. If you are super into mechanisms, this slide is for you. Now, what I do in our lab is I work with the bacterium that we get from our marine life, which is from nudibranch sea hares off of their backs. This bacteria is called Pseudoatromonas, or PL. It's a custom purchased plasmid that is unliving, so what we do is we transform it into living E. coli cells for mass production of the enzyme and then protein. Then we plate it on an agar plate. In our lab, we work with several mutants, but in my case, I work with a mutant called G489D, where the glycine was switched for an aspartic acid at the 489th base pair. Now, aspartic acid is very bulky, so in our case, we didn't think this would increase efficiency, but we did. And then with our procedure, we start from that agar plate after transformation, inoculate our culture media, and grow it overnight. Then we can express the protein in those cells, and then we lyse the cells to release that protein in our lysate, which, as you can tell in the picture in the middle, should be red if it's active. And then after that, we can add our reaction reagents, including those substrates. From there, we run it on LC-MS and hopefully see our hinokial product peak. There's a little intro to what I do for my side of a little bit of cancer research at Waynesburg University. Thank you so much.